Right. So today I'll be talking about a very important and so-called major transition in evolution, that from unicellular life to multicellularity. And I'm going to try to convince you about a very unconventional pathway of evolution where genes are not leaders but followers. So this would be the evolution of multicellularity, in which the genetic material kind of follows phenotypes which are induced by the environment. So the environment plays a key role, it changes the phenotypes, and then those phenotypes are fixed genetically. This is called the genes as followers mode of evolution. I'll be talking about multicellularity, so let's get into it. So the evolution of multicellularity was a major transition in evolution because it opened a key gateway towards more and more biological complexity. Life could become bigger, more complex, and hence more diverse. And multicellularity, unsurprisingly, has evolved many different times independently across all three domains of life. But what I would like to focus on is this particular thing, that multicellularity was invented on planet Earth by life among bacteria. We associate multicellularity with eukaryotes, or the so-called big organisms. I won't call them higher or lower, but and when I talk about multicellularity, I'll be focusing on bacteria, and I'll be focusing on the first step in this transition, which is from life with single cells to life with multiple cells in a cluster which are undifferentiated. So I'm not talking about differentiation just yet, but we can talk about it later. So because multicellularity evolved in the deep past, we have very little fossil evidence, and studying it is difficult. Thankfully, experimental evolution in the lab has come to our rescue. So what people tend to do is they start with unicellular, so the tube on your left that you see, which is more turbid and cloudy, it has unicellular yeasts, and it has been evolved for 600 days using artificial selection, and what you get are macroscopically multicellular yeasts. This is a very recent study, and this is the conventional way things are evolved in the lab using experimental evolution with microbes. Yeasts are also microbes. But there are two major blind spots in this approach. The first, particularly in the context of the evolution of multicellularity, the first blind spot concerns a very unwarranted focus on eukaryotes. I just told you that cyanobacteria invented multicellularity on planet Earth. So if you want to understand the origin of multicellularity, we should be thinking of bacteria, and gram-negative bacteria to be precise, because they are the ones who came up with multicellularity to begin with. But laboratory studies tend to focus on bigger organisms which are eukaryotes, and already which are very complex in terms of their genome organization and other things. And the second concern that I have with the conventional approach is their focus on mutationally driven phenotypes and not on environmentally induced ones. So mutationally driven phenotypes, particularly like multicellular phenotypes, can be difficult to obtain because you have to wait for the right kind of mutation, and that could be a long wait because mutations are rare. Beneficial mutations are rarer still. Okay, so I want to talk about environmental induction because there is a super powerful thing called phenotypic plasticity, which enables organisms to have the same genotype but exhibit multiple different phenotypes in different environments. For example, you see Daphnia here, where it forms this helmet-like protective covering when it's cultured with predators, and if the predators are removed, there is no such covering. It's in the same genotype. You can talk about butterflies which show different wing patterns and colors. It's the same individual but in different seasons. Or you can talk about the same individual which is simultaneously experiencing two different environments, one which is submerged and the other which is not submerged, and the phenotypes of the leaves would be very different. So this kind of phenotypic plasticity can be a very powerful force in evolution, which is what I would like to convince you about. So genes are important, but they can not only be leaders but followers in evolution. So what would happen if genes are followers, and what would happen if genes are leaders? So this is the conventional pathway. Let's talk about it in the context of multicellularity. So if genes are leaders, let's say this is a single cell, and it's being cultured in environment A. It's just one environment, and it would keep getting cultured in the same environment, and then a very difficult to acquire mutation would appear, which would then here change the color of the cells just to show you a change in the phenotype due to the mutation, but these cells will then stick together and multicellularity would originate, and this would keep happening, and then the propagule would be released and the life cycle can go on. There is no environmental induction, not a big role is played by the environment. The only role that the environment is playing is perhaps selectively enriching the multicellular phenotypes, but it's not giving rise to them. On the other hand, genes can also be followers. So think about this scenario where you have two different environments and some cells experience a change in their environment, and this change in the environment itself changes the phenotype. So now it makes them form a glue-like thing so that cells stick together, and now you've got multicellularity without the color change because the mutation has not happened. It's the same genotype but a different phenotype, and now after the phenotype has emerged, a mutation can happen which can then make this originally inducible phenotype a constitutive one. So in this blue cell, the phenotype has become constitutive now, and now if the blue cell goes to the other environment, to the original environment, which is environment A, it will still be multicellular. So now you have kind of genetically assimilated or fixed multicellularity, so that in both the environmental conditions you are multicellular. So yeah, this is a nice story. Does it really work? This is what I wanted to understand. So this process would be called genetic assimilation. That's what it's called in the formal literature. We have very few examples of a successful demonstration of genetic assimilation. And I wanted to ask if phenotypic plasticity can facilitate the evolution of macroscopic multicellularity, and that too in bacteria. So this was my seeding discovery as a second year PhD student. While I was doing my PhD with Sutit at IISER Pune, I discovered this. That if you grow E. coli, wild type E. coli, these are not engineered bacteria under habitual salinity, they grow as unicellular cultures. But if you grow them under high salinity, they don't grow unicellularly, or not just unicellularly, but they grow as a macroscopic filament-like thing. And these filaments can have more than 1 billion cells. So you don't really need to wait for mutations for something as common as an E. coli or something as simplistic as an E. coli setup. I would not call it a single cell. I would not call it an organism, because this calls for a re-evaluation of what we mean by an individual. So E. coli growth pattern or E. coli setup, maybe we can call it, for it to be multicellular you don't need mutations. So then we wanted to know if it was just a quirk of biophysics or some funny biochemistry going on, or was it an evolvable phenotype. And before that, we also wanted to understand if only E. coli was doing it, or many other bacteria do it. And what we found out was that this seems to be quite a common phenomenon. Both gram-negative and gram-positive bacteria tend to do this across habitual and high salinity. And by high salinity, I just mean 5% extra salt weight by volume. So 5 grams in 100 milliliters of your liquid medium, extra NaCl. Okay. What is more important is that this common ability seems to be highly conserved, because E. coli and Staph aureus here, they are gram-negative and gram-positive respectively. They diverged 3 billion years ago. So it seems to be a very highly conserved capacity. It doesn't manifest itself very frequently in front of us, but maybe we are not giving the right conditions, but the bacteria have somehow retained this capacity to grow in a very macroscopically multicellular fashion. So then we wanted to know if this was again just biophysics or biochemistry, or was it an evolvable phenomenon. So for that we do, for such things we tend to ask questions using experimental evolution, and that's what we did. We started with a single genotype which is represented by a single ancestral colony which is unicellular, or the genotype is unicellular in habitual salinity. We started 2 lines, S lines, which where S is for shaken, the tubes are shaken. So we culture them in a tube with high salinity environments, and we shake these tubes, we get these filament-like things which I showed you, and then we take a small propagule and then we wash it so that any planktonic or unicellular cultures that are present get diluted serially 100 million times, which means that if there were any residual cells which are unicellular, they would be washed away or selectively eliminated, and there would be an incentive to grow multicellularly. And we keep doing this again and again, but we did something more. We started something known as R lines. R is for resting, and the R lines don't shake, otherwise everything is similar, and same dilution and washing keeps happening. And we continued this for 50 days. The catch was that the environmental salinity reduced progressively, little by little. Okay, when that happened, this is what we observed. So there is the ancestor for your reference, and there is no environmental induction, so this is happening without any extra salt, so we expect unicellular growth. But in the selected lines, the growth was multicellular, and these are big globules which have each globule contains more than 1 million cells, and they are all alive. If you use fluorescent lines, they all fluoresce, and they are so heavy that they settle. We expected something different under resting conditions, because the oxygen gradient, is real and very dramatic, steep, because of lack of shaking. So these are R lines, which are resting lines. So we expect growth to be more, inclined towards the air-liquid interface, because that's where the most oxygen is present. And we really found out that, again, indeed, multicellular phenotypes are selected, but they tend to be at the top. So depending on the environmental conditions, we found multicellularity or its genetic assimilation, but the phenotype manifested itself slightly differently based on whether the tubes were shaken or not shaken. So then we found out that phenotypic plasticity, which was the original capacity of the ancestor to form multicellular clumps, facilitated the evolution of obligate macroscopic multicellularity, because now they did not need extra salt, they were doing it without the extra salt also. We wanted to probe more what was happening at the other levels. But before that, I must emphasize that this is different from biofilms. Biofilms are films, they are largely two-dimensional, they are not pillar-like or they are not globular, and they are, most importantly, ecologically speaking, they are facultative. My macroscopic multicellularity that is there is constitutively expressed, it's not facultative. So you don't need or you don't care about the environment, how much salt there is. Okay? So now we wanted to see what was happening at other levels of biological organization, because now perhaps for the first time, E. coli, which is a nice genetic workhorse in the lab, was giving us a macroscopic phenotype to work with. So it was a real treasure for us. So we wanted to probe more and what was happening to phenotypic plasticity at lower levels of biological organization. So we found out a very dramatic instance of plasticity at the level of single cells. So the E. coli, they are coliforms, so they have rod-shaped cells under habitual salinity. So this is the ancestor. But the cells become spherical. They appear circular in two-dimensional images when you give them high salinity. Okay, but we found out that in our evolved clones, something reverse happened. Instead of becoming more and more spherical, the cells became more and more tendril-like, more and more filamentous under high salinity. Okay, so that was very fascinating, because I'll just talk about a bit more about this. So this is what happened. So I'm just showing 10 clones here, 5 of S and 5 of R, and there is the ancestor for reference. Whenever we study phenotypic plasticity, the right ecological way of studying plasticity is through a quantitative way of measuring reaction norm slopes. So a reaction norm is a plot of a phenotype for the same genotype across two different environments. So here the two different environments are habitual versus high salinity, and the reaction norm slope, if it is pointing downwards, it just means that the cell perimeter, I'm showing everything in terms of cell perimeter here, which is a proxy for cell size. So the cell size is lower, okay, under high salinity for the ancestor. But what we found out was that the plasticity pattern or the reaction norms, they got reverted. We got reversal of the ancestral cell perimeter plasticity in 8 out of 10 lines. 2 other lines did not show this. This was important because when we mapped what was happening at the level of collectives, we got a one-to-one mapping of the genetic assimilation of macroscopic cell clustering or macroscopic multicellularity. So in the 2 cases where the reversal of cell perimeter plasticity was not found, we did not find macroscopic clumps without environmental induction. So there appeared to be a very direct connection between what was happening at the level of single cells and how phenotypes were getting manifested at the level of cell collectives, which were macroscopic. If we wanted to probe more, so we found out when we looked at these things quantitatively that plasticity evolved in opposite directions at different levels of organization. So when I talk about genetic assimilation, I say that the genotypes stop caring about the environment, they keep producing the same phenotype regardless of the environment after evolution, right? So this would mean that a slanted reaction norm would become flatter. Now there are multiple ways of making a slanted reaction norm flat. So this is the ancestral reaction norm which is slanted. You can make it flat by keeping this up so that you have a flat line up here, or you can make it flat by bringing the induced phenotype down so that you have a flat line like we observe here, which is what happened. So this is what happened at the level of individual cells. On the other hand, this whole thing went up instead of that coming down. Okay, so it was in opposite directions at the level of cell collectives. So plasticity evolved in the opposite direction at the level of cell collectives as compared to at the level of individual cells. This is a textual summary of the same thing. What I mean is that the evolved phenotype, which is non-spherical cells, have been expressed by the ancestor in the absence of induction. And at the level of cell collectives, the evolved phenotype, which was multicellular clusters, have been expressed by the ancestor in the presence of the environmental induction. This is a pictorial summary of the same thing. Now the point is, if I was just using the ancestor and I was just happy with this kind of result and extrapolate that if I fix multicellularity in some way, everything would become spherical because I am extrapolating from a correlation between being spherical and multicellular. I would be grossly misled because here multicellularity is getting evolved or assimilated by using tendril-like cells. Okay, so it means that plasticity has so much freedom to evolve in opposite directions across multiple levels of organization. So there may be a one-to-one connection or genetic connection, but those connections are super flexible. So then we actually looked at what those genetic connections were, and we found out that nothing was getting expressed on the surface of the cells, which was not really a surprise because the ancestor did not need a phenotype, did not need a genetic change, did not need a mutation, right? So without a mutation also, it had some kind of glue which could make cells stick in high salt. What this means is that you would get these assimilating mutations elsewhere in the pathway. They need not be glue mutations because you already have a glue. So these mutations happened in the peptidoglycan assembly machinery, which was in the periplasm between the two membranes or in the embedded in the inner membrane. And this is interesting because peptidoglycan assembly or it's the dynamics or speed of how much peptidoglycan can be assembled per unit time decides what the cell shape would be, which is unsurprising because we also found changes in the cell shape. So what we are getting is that then we also found that a single mutation, a single nucleotide polymorphism, a single SNP, a single amino acid change could lead to changes in plasticity, could canalize plasticity across multiple levels of organization. Plasticity in cell shape and plasticity in multicellularity. Okay, so this was interesting because the number of mutations that you need was just a very tiny number. It was only 3 mutations. On the other hand, if you do experimental evolution in the genes as leaders kind of mode or pathway, you would see tens of mutations, particularly over hundreds of generations, because you need precursor mutations, and then you need enabling mutations, and then you need enriching mutations to have a more and more refined phenotype. Here, the phenotype was already there. All that you needed was to make it constitutive, which is what a few number of mutations could manage. Yeah. So this is what we got, and then we wanted to understand what was the biochemical basis of plastic clustering, because there was some kind of glue, and we wanted to know if the glue was the same under both induced and uninduced conditions. And we found out that it was not. Under uninduced condition, there was the origin of a new glue. So what this means is that what these bacteria are doing is that they are keeping a macroscopic or a high-level phenotype, which is to have cell clusters genetically assimilated, but this is being brought about by different or contrasting lower-level phenotypes, which have different glues. So this actually elucidates the precise role that plasticity plays in evolutionary pathways. What happens is that plasticity primarily provides phenotypes at the outset itself without waiting for mutations, and these phenotypes can later be selectively favored. So it can speed things up, and it can be super flexible because the genetic assimilation of a higher level phenotype can be brought about by the expression of contrasting lower level phenotypes. So this is a summary of all the results which can be found in a recent paper that we had. Then I opened my own lab at ISO-TVM. So this is where we are at a very, very beautiful location, and this is where our lab is. This is my new group. we have just got our first PhD and master's student. So I'll be talking about, very briefly, I'll be talking about in 2 slides, some very exciting and recent results that we have got, which have been, driven by my master's student. So we want to ask now, we want to go from one species to multiple species to communities. So can plastic multicellularity facilitate the coexistence of antagonistic species of bacteria? So this is our question. We have 2 different bacteria, one gram-negative and the other gram-positive. There is a deliberate reason why we choose to work with these. Let's say Pseudomonas is red and Staph aureus is blue. It's a very common observation that when we co-culture them under habitual salinity, Pseudomonas drives S. aureus to extinction. There is a competitive exclusion. This is not just Stammel competition but also interference competition by injecting toxins and other diffusible molecules. So one bacterium kills the other. This presents a major conundrum because if you look at a very heavily infected cystic fibrosis patient, you will very routinely see PA-SA co-infections. But when you bring these things to the lab and try to co-culture PA and SA or Pseudomonas and Staph aureus in the lab, you would fail, mostly fail, because one will kill the other. So how are they coexisting or co-infecting our lungs in cystic fibrosis? So we have one solution to this problem, which is through multicellularity or phenotypically plastic multicellularity. So we say that maybe in the cystic fibrosis lungs or at wound sites, which is also a common site of co-infections, the environments are highly saline or high osmolarity environments. So it's not NaCl, you can also use magnesium sulfate and they would show the same thing. It's about salt. So maybe there is a local microscopic multicellular development that these bacteria are having, which is spatially segregating them. Maybe they are separated out, their ecologies are separate. So they are really kind of creating their own niches. I don't want to present it in terms of a very fancy word which is totally useless or unnecessary because it can be completely explicable in terms of modern synthesis. So I don't want to use the word niche construction, but they are constructing a place for themselves here. And what is happening here is something that we hypothesize. Did we get this? Well, we got something even more dramatic. We got spatial separation where the top layer is formed exclusively by Pseudomonas aeruginosa, which develops only at the top, and we got the bottom layer, which is more a pillar-like layer which starts at the bottom, which is exclusively Staph aureus. So then we wanted to ask, can such separation promote coexistence? And we found it very recently. This is like 3 or 4 days old result. So this is the fraction of Staph aureus, and within 3 days Staph aureus almost goes to extinction. So if we have this very stringent extinction threshold that the representation of Staph aureus should be 0.1% in the community, so that if you have a 1 in 1,000 sample, you would miss Staph aureus and you would call that extinction. Otherwise it's really difficult to establish when a bacterial species is extinct. So this is what we found, which is expected because Pseudomonas will drive it to extinction. But if we grow them under high salinity, where they grow like this, they don't go extinct for around 15 days. Okay, which could potentially explain why they coexist in our lungs where the environments are so hypersaline and so highly osmotic. That's all. Thank you. So I would like to acknowledge all my present and my previous institutes and my funding agencies to support very basic research which can potentially have nice applications also. Thank you.
Thank you, Yashar, for a nice talk. We'll just take one question because we are running out of time.
Okay, so you call it multicellular clusters, sometimes multicellularity, and you also mentioned phenotypic plasticity. So when we talk about multicellularity, usually we understand that there is some kind of division of labor in cellular function. Do you see any here?
I can show you slides which suggest that, but I don't want to claim it right now.
Because multicellularity, as we understand from traditional definition,
is
division of labor in the constituent cells.
That is differentiated multicellularity, but not foundational multicellularity, which
so that you haven't qualified.
That's why in my first slide itself I say that this is undifferentiated multicellularity, which is foundational. So
But the Nature paper says multicellularity.
It does say multicellularity because it is a kind of multicellularity. It is foundational multicellularity. It is, there is no doubt that they are multicellular.
Just one
So,
quick question.
Yes.
This glue that you find here,
Yes, yes.
is it biochemically different than the glue that you find in biofilm?
Very different. Yes, very, very, very different. Yeah, yeah.
Nice talk.
So,